It's going to be slightly different because I'm not going to talk about the work that we do in my lab at all, but if anybody's interested in hearing about it, I'd be more than happy to talk to you about it. I'm going to talk to you a little bit about what I see as the biggest obstacle to making this final progress in cancer, and we'll talk about what I mean by final progress in 1 second. Because I thought that's what would be most appropriate in this setting. And here is a picture of a human brain, a slice through a human brain, and in that brain is a cancer. This is a glioblastoma. And glioblastoma has particular relevance, I think, to this discussion because it's probably the brain tumor that we know the most about in terms of its molecular foundation, where it comes from, and how it develops into the kind of aggressive cancer it is. But it's also the cancer for which there has been little advance in cure since radiation therapy was introduced over 60 years ago. So 60 years ago, with surgery and radiation therapy, patients lived about a year. 60 years later, after an enormous amount of expenditure and money and effort, explosions in just knowledge about the molecular foundations for cancer, and in particular this cancer, patients still live only about a year, maybe 14 months. And the question is, why is that so? And that's really what I want to talk about today, because I think that's what we were after here. So let's start, with what is cancer. So here's a tumor that was taken out of a person. There's a human hand to give you an idea of how big that tumor is. And I put it up there for several reasons. I think it very simply illustrates several things fundamental to cancer. One is that cancer is dysregulated growth. It's dysregulated and it's disorganized. I mean, there's no sort of recognizable tissue structure there. And I want you to also remember how red it is, because cancer is incredibly vascular, and that's very important. And currently, what we do is we cut them out, and then we expose what might be left in the body to radiation and chemotherapy. And the goal is to inflict enough damage to DNA, to their metabolism, to the machinery that's necessary for living every day and dividing, so that the cancers stop growing and hopefully they die. The problem is that approach isn't always successful. I just told you, people with glioblastoma don't survive. And in fact, it's often incredibly toxic. And so the goals of cancer therapy, are to improve the cure rates and to improve the quality of survival. And there are several things that are necessary in order for that to be achieved, I think. And that's an understanding of cancer biology. And in particular, we're going to talk about what I think might be among the last frontiers in that. Faithful animal models relative to what I'm going to describe, and then appropriately designed clinical trials, which actually will get to the policy part and sort of a shift in how we think about cancer clinical trials. But to start with, I want to talk about what is cancer. And when most people talk about cancer, they talk about the cancer cell. And in fact, cancer research has been dominated by this view of cancer that is cancer cell centric. And there's a long history of thinking about cancer in this way. And I want to highlight some of the important findings. And I want to also emphasize how long research has been focused on what is cancer. This is only the last 100 years, but it's only the last 100 years of actually centuries of people thinking about cancer. But just about 100 years ago, among the most important cancer cell focused findings was really made in sea urchins by Theodore of Bovary, who was watching sea urchin development, had noticed that there were anomalies of sea urchin development, and that within the cells of the anomalous structures, there were abnormalities in chromosomes. So remember, this is before the discovery of DNA. There's no sense of genes being responsible for, any part of biology, but there is a knowledge that chromosomes have to be replicated, and they are equally divided between daughter cells under normal circumstances. And here's an abnormal circumstance, and there's abnormal chromosomes. And he actually postulates in 1914 that chromosomes will be regulating growth and that cancer could involve abnormalities in chromosomes. Time goes on, and a number of epidemiologists, including Armitage and Dahl. Sir Richard Dahl was knighted for making the connection between cigarette smoking and lung cancer. But they're just studying the frequency with which cancer occurs in human populations. And they're noticing that cancer increases with age. And in fact, it increases as a fifth or a sixth order of age. And they actually postulate that in the cancer cell, 5 or 6 things have to happen to turn it from a normal cell into a cancer cell. And they say that those things will involve mutations, changes in chromosome structure, and changes in tissue structure, and we'll come back to that. But I want to point out that just recently, in the last 10 years or so, Bob Weinberg at MIT determined that it took about 5 mutations, 6 mutations, depending on where you start, to take a normal cell and turn it into a cancer cell. The careful observations that these guys made just from epidemiology accurately predicted that. In 1960, finally, the first chromosomal abnormality associated with cancer is described. It was described by 2 people working in Philadelphia and became known as the Philadelphia chromosome. And it's something that you find in leukemias, and we're going to come back to talk about it again. Soon afterward, 2 other chromosomal abnormalities that are common in lymphomas were described. But here it was, finally realizing what Bavaria had predicted, that in a cancer cell, chromosomal abnormalities would drive the abnormal cancer. And then among the most important findings was in 1970, when Alfred Knudsen, basically working based on what these guys and Nordling had predicted, looks at a pediatric cancer, retinoblastoma, which occurs in 2 forms. One is sporadic, and the other is in families that have histories of retinoblastoma. And postulates, doesn't define the basis, but postulates that there are genes within cells that function as what we now refer to as tumor suppressors. He didn't use that term, but things that will block the development of cancer. And that because there are 2 alleles for each gene, if you mutate both alleles, you lose tumor suppressor function and cancers develop. That's the basis of what you may have heard as the 2-hit hypothesis. And he went on to describe how you could inherit one of those mutations. So you're all familiar with cancer predisposition syndromes now. BRCA mutations in breast cancer, RB mutations in retinoblastoma, P53 mutations in all sorts of cancers. Or you could have both of those things happen somatically in a somatic cell. By bad luck, basically, and that would determine a lot about the age of onset of the cancer, the frequency of the cancer, and the multiplicity of the tumors. So that's where we got the idea of tumor suppressors. And then just a few years later, Harold Varmus discovers the first oncogene. It was the SRC gene, which had been usurped by the Rous sarcoma virus. So the oncogenes are mutations that are gains of function. Things become dysregulated and promote growth. The tumor suppressors are loss of function. You lose the ability to protect against cancer. The end result of all of this 100 years of research was the focus on the cancer cell as the source for everything that's wrong in cancer. And it elevates this sort of, sorry, I'll keep doing that. It elevates this expectation that if you understood the cancer cell, you could treat what was wrong in the cancer cell and you could make the cancer go away. And so we take the example of this Philadelphia chromosome because it is the single clearest example of what's right and what's wrong with this. So the Philadelphia chromosome takes part of chromosome 9 and part of chromosome 22 and brings them together to create a completely abnormal protein known as BCR-ABL. BCR is for the breakpoint cluster region, and ABL is the name of a tyrosine kinase. And it creates a tyrosine kinase that's completely dysregulated. It's constitutively active. It's always on. It's always promoting growth. And it's an ideal therapeutic target because it simply does not exist normally. So if you had a drug that could target that activity, the only cells affected by it would be the cancer cells. It's perfect. And Novartis, a giant drug company, had a drug known as Gleevec or imatinib that actually could block this kinase. Turns out it can block some others, but it mostly blocks this kinase. And in fact, the BCR-ABL was present in chronic myelogenous leukemia. And Brian Drucker, in 2001, reported the use of Gleevec in treating patients with chronic myelogenous leukemia. And the astounding finding, 53 out of 54 patients responded to this drug. So remember, this is not like cytotoxic chemotherapy. It's not radiation therapy. This is a tyrosine kinase inhibitor. It binds to this protein. It blocks its catalytic activity. And 53 out of 54 patients responded to it. It seemed remarkable, and it was remarkable, except that there were many things that made this almost unique to chronic myelogenous leukemia and with little applicability to other cancers. Among those things is that most cancer is not the result of a single genetic abnormality. Just like Armitage and Dahl had predicted, it's the result of multiple abnormalities. And in fact, glioblastoma has hundreds to thousands of mutations. Not all of them are important for its growth, but many of them are. And so it's not possible to think about targeting a single abnormal function and getting a response. CML is almost unique in this, in that it's driven by a single abnormal event. The other thing that was not clear, and actually only until a couple of years ago was not clear, it was postulated but not known, and that is there is no single cancer cell in most cancers. So CML happens to be a little bit unique in that it truly is clonal, meaning that if you hit that one target, you basically take care of all the cells in the cancer. But that's not how cancer actually is. So here's real data, not mine, from a leukemia at the time of diagnosis. There are 9 different clones in this leukemia in the single biopsy specimen. They differ slightly, maybe. There's one primordial clone, but it gave rise to 8 other clones. And the problem is you take a drug like Gleevec and you treat something that's present in one of these, and maybe you take care of that one clone, but there's all the other clones there to grow back, to dominate at the time of recurrence. And it's not possible in most cancers to think about a single target. And you may ask, well, why can't you target something that's in the primordial clone? And that's a great question. And among the answers to it is that just like in normal development, there are genes that are important for development but then are no longer important for maintenance. There are genes that are important in the genesis of tumors that simply aren't that potent as targets once the cancer is formed. And so it's difficult to think about targeting the cancer cell, and it's difficult to think about targeting a single target. We'll come back to that in a second. So what are your alternatives? Well, you can think about the tumor tissue. So just remember that Armitage and Dahl had postulated that disruption in tumor tissue or tissue structure would also contribute to cancer. There's also a long history of thinking about this. It's actually a very long history of thinking about this. But in the modern era, there's at least 30 years of really solid data to suggest that the whole story is not about the cancer cell. So consider this for a second. In 1981, Stewart took cancer cells, injected them into developing mouse embryos, was able to trace the lineage that derived from those cells. And those cancer cells, in the setting of that normal development, gave rise to normal tissue. So that's remarkable, right? I mean, those cells gave rise to normal cells. In the mid-90s, that Rous sarcoma virus that Harold Varmus had discovered was injected into developing chicken embryos. It got integrated into the genome. This is a constitutively active oncogene, but it didn't cause cancer unless they generated some inflammation. And they could generate the inflammation anywhere because they got it incorporated into the genome. So, and that inflammation could be just taking a needle and moving it around a little bit. As soon as there was tissue disruption, as soon as there was inflammation, tumors formed. It wasn't just about the oncogene and the cancer cell. And then there's a whole bunch of studies like this. I'm highlighting this one because I think it's just a beautiful study. Maffini took newborn mice, female mice, and took out the breast epithelial cells. These would be the cells that would give rise to carcinomas. And cultured them, kept them alive, kept the mice alive. When the mice were about 50 days old, he irradiated the mammary fat pads. This is the de-epithelialized mammary tissue. Or irradiated the cells in culture, or not. And then put the cells back into the irradiated or non-irradiated fat pads. It was only irradiated fat pads that gave rise to tumors, and it didn't matter whether the epithelial cells had been irradiated or not. Take normal cells, put them into irradiated fat pad. You got tumors of the epithelial cells, not of the fat cells. You could take irradiated epithelial cells and put them into non-irradiated fat pads, and they didn't make tumors. The normal tissue structure was inhibitory for tumor formation. Disrupted tissue structure was permissive, even promoting of tumor formation. So maybe you can target the tumor tissue. So you'll recall that that giant tumor I showed you was really red. And I purposefully chose that picture because of how red that tumor was. And many of you may have heard of Judah Folkman, and also in the 70s at about the same time that tumor suppressors and the oncogenes were first being described. Judah Folkman was a pediatric surgeon, in Boston. And what he noticed is that when he took tumors out of kids, they were always incredibly bloody. And he postulated at that time that tumors, in order to grow, had to stimulate blood vessel growth. And that you could think about treating tumors not by treating the cancer cells, but by blocking blood vessel growth. So he single-handedly really gave rise to this field of the study of angiogenesis, the formation of blood vessel growth. And I want to distinguish pathologic angiogenesis from normal angiogenesis. And the idea that you could develop anti-angiogenics as cancer therapeutics. And today, we know an enormous amount about angiogenesis. And there are 60 or 70 molecules that regulate angiogenesis under normal circumstances. And among the most potent of those is vascular endothelial cell growth factor, or VEGF. And it's made in huge quantities by cancer cells like those glioblastoma cells. They just make tons of it. And they secrete it, and they stimulate blood vessel growth at an enormous rate. And their growth is dependent upon that blood vessel growth. And so targeting angiogenesis seems like a fantastic way to get around all this complexity of tumor cell heterogeneity and all the different molecular events. Unfortunately, it doesn't work that well. So there are a number of drugs. Bevacizumab or Avastin is an antibody against VEGF. You can give it to humans, and you can bind up all the VEGF. And you can see, here's the survival advantage for colorectal cancer. Here's without VEGF antagonist, here's with VEGF. So it definitely has a survival advantage, but it's really slight. And the reason it's really slight is very similar to the problem with Gleevec. There are 60 or 70 different molecules that can regulate angiogenesis. And it's not just about the endothelial cells, which are really the target for VEGF. It's about pericytes, and in the brain it's about astrocytes, and it's about microglia, and it's about bone-derived stem cells for all of these cells. All of these things contribute to angiogenesis, and they all regulate angiogenesis. And there's more than enough ways that a tumor can get around the depletion of VEGF. And so together, this starts to highlight what the problem is. So the current state of the art, we think about single targets in the cancer cell. We think about single elements of the cancer microenvironment, and we conduct clinical trials to evaluate new drugs to target these things in the following way. We take a group of patients, let's say it's 100 patients, and we're going to test a new drug, something like similar to Gleevec, let's say, or something similar to Avastin. And we give the drug to 50 of them, and we give something different to 50 of them. And we defend our experimental design as good because we say all the patients are the same. And we demonstrate that they are equally distributed in age and in prior treatments and in sex and whatever other parameter you might think is important. And invariably, the results of those trials are pretty poor. And they're pretty poor because all of these things are wrong. It's not a single target. It's not a single cell. It's not a single element of the microenvironment. And everybody's different. And all of their cancers are different. And so the problem with the state of the art, the thing that I think would be the focus we're thinking about tonight, is that cancers are incredibly heterogeneous. And that unless you embrace that heterogeneity and you incorporate it in how you think about cancer biology and how you think about treating cancer, there's little hope of, I believe, in curing those glioblastoma patients. And so what I think, God, it's ridiculous, is the obstacles are understanding how to deal with heterogeneity. And I want to finish with what we're trying to do in clinical trials, not in the labs. We can talk about the lab and research if you want later. But in clinical trials, I want to talk about what it is we're trying to do. So currently, as I described, you would introduce a new drug into clinical trial by treating a group of patients who had already recurred after everything that you had available to give them. Let's say there were 40 patients. And you would treat them all with this new drug that you had determined in laboratory setting, which would involve presumably both in vitro work in a dish and in vivo work in a mouse. And the mouse would be an inbred strain of mice, and it would use a single model for, in this case, brain tumors, let's say, that you had developed in the lab and maybe even injected into the brains of those mice. And you would give the 40 patients that drug. And I can tell you that after years and years of clinical trials that are constructed in this way, chances are no more than one, maybe two patients would demonstrate some response to that drug. Rarely there's something that's a little bit better than that. And in general, the conclusion is that that trial was a failure and everybody moves on to the next drug. And I think the problem with that is the way that, in the way I've been describing, the way that we think about cancer and the way that we think about clinical trials. I would propose that what needs to happen is a shift in thinking about this to the following. If you said for any drug or any treatment, it doesn't have to be a single drug, that what I want to do in a first phase of clinical trial is determine what distinguishes that patient who responds from those who don't. And so even if there's one patient who responds, if your goal is to figure out what distinguishes that one patient from the other 39, you have a successful trial. And what I mean by distinguishing it is being completely agnostic about what might distinguish them. So genome-wide distinction, proteome-wide distinction, biology-wide distinction. And you know, the problem with, and we're trying to get a trial like this open, and I'll tell you that among the problems are that, one, people aren't used to thinking about clinical trials this way, and even if the idea excites them, they're not quite sure how to construct it, how to even approach the statistical analysis for it, for instance, because the statistics around clinical trials use these very standard models. But maybe more importantly is the laboratory support for it. So I'm going to give you a real life example. So right now we're involved in a clinical trial in a consulting way for breast cancer that involves an antagonist for a molecule CXCR4, which is one that's a big focus of our laboratory research. We know that CXCR4 is important in cancer. We know that in multiple cancers, all brain tumors, that CXCR4 function is abnormal. It's different than its normal function. And it's different in its normal function because the primary mutations, those things in oncogenes and tumor suppressors that are changing the biology of the cancer cell, secondarily alter the way that CXCR4 functions. It's something that you just don't find under normal circumstances. And among the things that happens is it changes how much time CXCR4 spends on the surface of cells and how it engages signaling molecules that it can access from the surface. And so if you wanted to study response to the drug, you might want to know something about where CXCR4 is in the cell, how much time it's spending there, and whether it's engaging these signaling molecules that we think make its function different. But the standard approach isn't to do anything like that. It's to simply say, most commonly on the basis of gene expression profiling, how much CXCR4 is there in a patient. Not how much protein is there. Not is the protein intracellular on the surface, not is the protein engaging signaling pathways, not how is it interacting with the other primarily mutated pathways. And the practical problem is, who's going to make those determinations? I think they should be made, but my lab's not about to do it. It's not what we do. It's not what I'm funded to do. The clinical trials don't support funding for that, and the drug companies don't tend to want to spend money on that. And so from a policy point of view, what there needs to be is a big shift in several things, in my view. They involve what we establish as the objectives for clinical trials, and then how we support clinical trials. Because it shouldn't just be about finding the magic bullet, as you said, the new drug that's going to cure everybody. It should be about identifying those people that respond to drugs. It will require changing how you staff labs to do these things. So it's not, I don't think it's going to be possible, for instance, to have a senior level scientist who needs to get grant support to run a lab like this. This should be a hire. This should be a staff position. It should be somebody whose job is guaranteed as long as they can continue to support clinical trials in this way. And that sounds like a simple idea, but you know, go talk to a department chair about hiring somebody who's not going to be bringing in grant money to support what will be expensive work. So there are policy aspects to this that I hope seem important, because I think they're really important, that would support this kind of shift, which I think is what is actually required if we want to make the kinds of advances in cancer that remain to be made. And that's all I'm going to say.
Any questions?
Yes?
So it seems like what you're talking about is basic versus applied research. There's something infrastructure just supporting the stuff, as opposed to, like you're saying, the magic bullet to actually get it done there.
Yeah, so applied science but it requires some sort of fusion of, you know, sort of what we would refer to as, you know, what we've always pursued in science, because it's not going to be possible to know beforehand, I don't think, what the signatures are going to look like. So it's going to require scientists who are really senior level scientists, I think, to be tackling to be tackling series of questions, right? It's not going to be a career path like we usually think about it. It's going to be problem after problem after problem after problem, but each approached, you know, in the way that a scientist would approach a problem. There's something to be understood, there's things to be learned, and there'll be adaptations that need to be made along the way, and how you think about it and what experiments you do. And I don't know if I'm making myself clear there, but you know, I see this as a fusion of what we've always done, but now also as a, you know, almost like a core, except a very, very dynamic kind of core.